project title is Preparation and Characterization of Yogurt-like Product Enriched with an Exopolysaccharide Producing Bacillus species RT1. Yogurt is a fermented milk product obtained by fermenting milk with lactic acid bacteria. The most commonly used strains are Streptococcus thermophilus and Lactobacillus bulgaricus, and yogurt do have many health benefits. The problem with the commercially available yogurt is the use of additives in order to enhance the nutritional value of the yogurt. The overuse of traditional starter culture reduces the bacterial diversity in natural reserves. Hence, our aim is to prepare an improved yogurt using exopolysaccharide producing bacteria because exopolysaccharide producing bacteria is generally used in food industries to enhance nutrient and texture in food products. The objective of this project is to isolate and characterize exopolysaccharide producing bacteria from various dairy sources to prepare yogurt-like product using isolates to analyze the characteristics of the prepared yogurt-like product. And this is our workflow. Various dairy samples are collected and EPS producing bacterial species are isolated. The milk is fermented with the isolates in order to obtain the yogurt-like product. The isolate is selected based on its enhancement in nutrient content in the yogurt. The product's shelf life is analyzed and the isolate is identified using 16S rRNA sequencing. EPS producing bacterial colonies are screened from a pool of colonies based on its slimy nature and biofilm formation in MRS broth. In order to confirm the presence of exopolysaccharide, capsule staining is performed for the 8 isolates. In order to prepare the yogurt-like product, the milk is heated at 90 degrees Celsius for 30 seconds and cooled suddenly at 42 degrees Celsius for 10 minutes. Now the isolates are inoculated as starter culture and allowed for fermentation until it reaches a pH of 4 to 4.6. The product is observed for its texture and appearance. The yogurt with whey casein separation are excluded. The following characteristics are analyzed for the prepared yogurt-like product: total titratable acidity, protein, carbohydrate, fat, solids not fat, and microbial load. These analyses were performed based on the FSSAI standard protocols and the strain was selected based on the guide values given by the FSSAI. This is the summary of the selection process. Now, stability analysis was performed for the product prepared using the selected isolate, and it was found that the shelf life of the product is up to 2 to 3 weeks. In order to identify the isolate, 16S rRNA sequencing was performed. The isolate was found to be Bacillus subtilis. Now, there lies a question that whether Bacillus subtilis is used as a probiotic. So, we did a literature study and we found that some strains of Bacillus subtilis is known to have probiotic effects, and it's also used in making fermented milk products. Hence, our future work is to analyze the probiotic potential of our isolate.